रोहित भाई, रेवांगिनी जी को को होस्ट बनाइए।
डन सर।
सईदा जी को को होस्ट बनाइए। रोहित जी, सर ने ज्वाइन कर लिया है, सर को कोस्ट बनाइए। भारद्वाज साहब ने ज्वाइन कर लिया।
ओके सर।
नमस्कार सर। जय श्री राम।
जय श्री राम। नमस्कार सर, आवाज आ रही है?
जी, मेरी आवाज आ रही है सर। बस 5 से 7 मिनट में स्टार्ट करते हैं सर।
श्योर सर, आई एम हियर ओनली।
ठीक है,
रेडी
थैंक
सर।
यू सो मच, थैंक यू।
ओके, नमस्कार नमस्कार।
रोहित जी, अजय को को होस्ट बना दीजिए। डॉक्टर रेवांगिनी, आप सुन पा रहे हैं मुझे?
यस सर।
200 प्लस पार्टिसिपेंट्स ने यहाँ ज्वाइन कर लिया है और
ठीक।
117 पार्टिसिपेंट्स यूट्यूब पे ज्वाइन किया है। आई थिंक 350 कैंडिडेट्स आ चुके हैं। ठीक है, स्टार्ट करते हैं। रेस्ट ऑफ द कैंडिडेट्स आते रहेंगे इन बिटवीन। ठीक है,
ओके सर।
ठीक, आप रेडी हैं?
यस सर।
210 पार्टिसिपेंट जूम पे भी है अभी। ठीक है, 350 प्लस पार्टिसिपेंट्स है, रिस्पेक्टेबल है। चलिए स्टार्ट करिए प्लीज।
ओके सर, थैंक यू सर। Namaskar to one and all present here. I am Dr. Evangini Ranjha, Director HR at AgriMeet Foundation Bharat, and your host for today's session. On behalf of AgriMeet Foundation and our visionary leader, Dr. Ankur Sharma sir, I extend a warm and heartfelt welcome to all the participants to Ankuran 2026, 30 Days International Summer School Cum Training Program. Ankuran has been envisioned as a platform to connect young minds with cutting edge scientific innovations, emerging agriculture technologies, and renowned experts who are shaping the future of agriculture and allied sciences. Today, we are privileged to host one such eminent personality whose contribution have significantly advanced pulse research and crop improvement at both national and global levels. It is my great honor to introduce our esteemed speaker, Dr. C. Bhardwaj sir, Principal Scientist, ICR IARI, New Delhi. Professor Bhardwaj sir is a distinguished legume breeder with more than 28 years of experience in chickpea breeding and genomics. An outstanding alumnus of ANGRAU Hyderabad and JNKVV Jabalpur, he has developed 23 high-yielding and climate-resilient chickpea varieties. Among his landmark achievements is the development of world's first marker-assisted bred chickpea varieties for drought-tolerant Pusa chickpea 10216 and wilt resistance Pusa chickpea Manav, establishing India as a global leader in genomic assisted pulse breeding. His scientific excellence is reflected in more than 209 research publications in reputed journals, with an H index of 41 and over 6,200 citations. His remarkable contributions have earned him numerous national and international recognitions, including the prestigious Grigor Johann Mendel Award, Professor M.S. Swaminathan Award, Dr. G.S. Khush Plant Breeding Award, IIMT Breeder of Excellence Award, AgriWatch Research Recognition Award, AgriSet Partnership Award, and AgriVision Eminent Scientist Award. Professor Bhardwaj sir is a fellow of Springer Nature and several premier scientific societies and served as an honorary adjunct professor at leading institutions in India and Australia. He has led several high impact national and international research collaborations with organizations such as Bill and Melinda Gates Foundation, ICRISET, DBT, DST, and CIMIT. A dedicated mentor and academic leader, he has guided 14 PhD scholars and contributed to the training and evaluation of more than 60 doctoral students in agriculture sciences. Currently, he leads the chickpea breeding and molecular breeding program and serves as a professor of genetic and plant breeding at ICR Indian Agriculture Research Institute, New Delhi. Sir, your achievements and contributions are so vast and inspiring that they truly cannot be covered in a single introduction. Your journey continues to motivate thousands of students, researchers, and young scientists across the globe. So, on the behalf of entire AgriMeet Foundation family and all the participants of Ankuran 2026, I extend a very warm welcome to you, sir, and express our sincere gratitude for sparing your valuable time to join us. Now, without further ado, I respectfully invite Dr. Bhardwaj sir to address the gathering and share his valuable insights with us. Sir, the platform is all yours.
Oh,
Please, sir.
are you able to hear me?
Yes,
Yes,
डॉक्टर
sir.
साहब, yes.
Only I think that I'm having some problem with the video. It's not starting. But
रोहित जी,
I'll go ahead with my presentation.
Okay, sir.
Why it is turning round and round? Oh my God. Nothing is happening. It's just going. Can I just give me 1 second for log out and log in?
Yeah, rejoin, sir. Just
No,
rejoin.
I think it is helping. It is sharing. Zoom is not generate a crash report center. It is visible now, sir?
Yes,
Yes,
sir.
sir.
Yes, sir. Yes, doctor.
PT mode. Yeah, it's working, sir. Fine.
It's working fine. I'm really sorry that you are not able to see me, which I am missing myself. But thank you for that excellent introduction, and thank you, Ankar Bhai, that you always make it a point that I'm on your program. Today, I'll be speaking on genome-wide scans and pyramiding of Fusarium oxysporum sisor alleles for wilt resistance in chickpea. Before I start my lecture, I really place on record heartfelt thanks to my co-authors, my students, and my co-fellow researchers. Dr. Neeraj Kumar, who is a senior scientist working in chickpea program. Dr. Jorben J, scientist at BHU Varanasi. Yogesh Kumar from IIPR Kanpur. Dr. P.K. Jain, Pradeep Kumar Jain from NIPV at New Delhi. Priyadarshini, who was a student here and now is a scientist at IGFRA. Apurva, Raupreet, Umashankar, who are my students working on the same lines and evaluating lines. Professor Manish Rurkiwal, who is now working with Reliance Industries Limited and earlier with ICRISAT and Khalifa Center of Biotechnology, Dubai. Professor Rajiv K. Varshne, who was principal scientist and genomics lead at ICRISAT and now director, Food Futures Institute, Murdoch University, where I'm also adjoined. Now, we do understand that chickpeas are one of the most important nutrient-dense crop, particularly to the vegan and to the vegetarian population. Nevertheless, not only they're packed with proteins, they're packed with high digestible proteins, but at the same time, they're source of vitamin B6, folate, vitamin C, a good amount of soluble fiber, and that's why they're said they're diabetic friendly. Either you're looking at hypocholesteromic or digestive, promoting heart health, controlling blood sugar levels, supporting amylose, beta, cytosterol, or raffinosis. So it's a resistant starch, and therefore they say that at least 50 to 100 grams of your fiber and protein should come from chickpea. Of late, where the vegan meat or the vegetable meat industry has grown from $124 billion to the current $284 billion in the last 2 years, recording a 400% increase. Chickpea protein is, or the chickpea protein colloids form one of the most important aspect. Though we have a larger amount in case of soybean, but then of its 40%, 20% is non-digestible, and of the remaining 20%, it does not give a meat-like flavor when used in its colloidal form, forming a leathery texture, and is allergenic to many people. Pea proteins give a green color to the protein colloids, whereas Kabuli chickpea has colorless protein colloids, has meat-like what you call as organoleptic properties, has an excellent amino acid profile next only to egg, and therefore is called as vegetable egg. You can imagine amount of dietary fiber or protein, and it also has very good amounts of riboflavin. Now, if you look down from that of 9.21 million hectares, we have gone down, we have gone up to about 14 million tons. From 8.88 million tons, chickpea now commands almost 14 to 18 million tons. It wavers around. From zero point 0.9 tons per hectare, where the world average is, to the present chickpeas production average, that is productivity of 1.2 tons per hectare, chickpea has traveled long strides due to the effort of numerous chickpea breeders, due to policy planners, and most importantly, the farmers' trust in this resilient crop. India is the world's largest producer of chickpea, and the parody is that we are also the largest consumer of chickpea. Pulses, nevertheless, are the rulers of semi-arid tropics, where rice and wheat can't come. People grow pulses, and they're always pushed to terminal soils without fertilizers or without totally dependent on rainfall. Therefore, where the livelihood of farmers who are small or very small farmers in these semi-arid tropics, chickpea and other pulses form their backbone and the strength to overcome the difficult terrains that they pass through. We have an excellent group working on chickpea improvement, whether it is breeding, agronomy, plant genetic resources, plant protection, or plant extension. That is the All India Coordinated Research Project on chickpea, now rechristened for the last 2 years on Rabi crops, All India Coordinated Rabi Improvement Program, where you have all the Rabi crops, and Dr. Sailesh Tripathi is the coordinator, and the whole of the country is divided into 5 zones consisting of various states. But why I wanted to give this prelogue is whether it is Central and South India, whether it is Northeast Plains, Northwest Plains, if you look across them, one of the dominant essential priority trait always has been wilt. Whether it is short duration, high temperature, and drought tolerance in the Central and South India, but it should be resistant or tolerant for a cultivar to be if it has to be released in that zone. In Northeast Plain zone, where we look at late zone varieties with medium to short duration, high temperature, but wilt and botrytis grain mold. If you are looking at Northwest Plain zones, where I am located, including the plains of Jammu and Kashmir to that of Punjab, Haryana, northern Rajasthan, western Uttar Pradesh, then apart from cold and Ascochyta blight, wilt. That is, wilt is a underlying phenomenon. So this is one of the most important numero one biotic stress. Of course, there is heliothis, and of course, we don't have what we call as the normal breeding approaches than for anti-feeding activities like breeding for greater pubescence on the pods or on the leaves or tuberculated what you call as seed coats. But then one of the major thing is Fusarium wilt, and the problem with Fusarium oxysporum forma species Caesarea is there has been a changed race and pathotype scenario from Professor Nene Saab's of ICRISAT, where he has clearly identified 6 fork alleles and 6 fork races, to that of numerous pathotypes given by Professor Dubey, now Honorable Vice Chancellor, Bihar Agricultural University, Sabor. So we have good tolerant sources. I don't say we don't have, but they are in the secondary gene pool, which are a multiple resistant, and crossing is difficult. And most important thing, as I indicated, is that this particular pathogen has evolved into numerous biotypes. Favorable conditions are 24 degrees with an inoculum load of 104 to 105 micro or macroconidia. It's a soil borne and has a huge amount of survival till 6 years. The symptoms are very characteristic, that the drooping starts from the top and proceeds down. This happens because basically the xylem gets clogged by the fungi, and therefore there is a black discoloration. Therefore, drooping starts from top to down. Now, the classical Professor Nene Saab's this thing was that we had 3 and 4 prevalent in the Northwest Plain zones, to 2 and 4 in the central zones, to 4 and 1 in the southern zones. But then, with the latest work of race profiling and molecular diversity analysis done by Professor Honorable Vice Chancellor S.C. Dubey sir. Now this concept has changed, and then they have actually found that it has now bifurcated it 70 isolates, with almost all the isolates found in a mixture at major places with major isolates. For example, in the central India, of the fork race 2, it is basically the Sihor isolate and the Jabalpur isolate, which we call as 11.5 and 7.7. To that of Northwest Plain Zone, where you have the isolate fork 4 with 2, that is Delhi isolate and the Ludhiana isolate. Similarly, you have the Dharwad isolate and the Hyderabad isolate in the south zone, to that of the Pantnagar isolate and the Kanpur isolate. So you have new differentials for each of these, but then how do you understand with such what you call as isolate diversity? So Professor Dr. Jorben, who is now a scientist at Banaras Hindu University, whilst he was here, developed an excellent hydroponic method. Why I call is a novel screening method is that we have for wilt fork alleles, but then he developed along with Apurva Rao and standardized it basically for pathotype screening. They have a basic rapid and reliable screening, because you can actually evaluate against the targeted isolates or a mix of isolates at a faster pace in hydroponics. And with fewer of the lines that have been identified, you can go along and do it microplots. The time my late friend Srinivasa has helped in this. And then those few lines, or those few breeding lines, which are totally resistant, you can start taking them and evaluating at multi locations. And this was an idea how we could actually build into an hydroponic based screening approach, where almost we could have screened a large amount of lines in the hydroponic setup, which is actually patenting this one, where we have used what we call as simplified techniques such as just normal tubs, perforated polygalactourinate sheets, cotton swabs, and of course, pure cultured isolates. So we had the Delhi and the Ludhiana isolate. We had the Jabalpur and the Sihor isolate. So against each zone, we had kept almost 2 dominant isolates. We have transferred 7-day same size old seedling plants, and we maintained a concentration of spores of 3.2 into 10 to the power of 6 conidia per ml. So specific pathotypes were added, and similarly we had a control. And then disease assessment was done on a special scale developed by Srinivasa, where the scoring was done on a 6-digit scale that went from 0 to that of 5. And we started looking at, and we also calculated the disease index, based on the scoring of the whole plant and the parts. And what we have basically done is we have actually developed a large number of introgression lines, which I'll be discussing in the last part of the lecture. But you see that against all the lines, we got a beautiful disease score. And at the same time, we also screened certain land races. For example, this is Icada landrace chickpea cultivar that is obtained from Czechoslovakia, had a varied reaction, whereas Latvia had a highly resistant for race 1, 2, and 3. BGD 112 showed complete resistance, and similarly certain introgression lines of the marker resisted breeding. We didn't stop that. We had done some histochemical studies, and in the susceptible lines, you'll see that the infected xylem was totally clogging the xylem, whereas in the normal xylem, the resistant plants, there was no clogging, and hence there was a perfect function. We had Varshne there. So what we were interested, once we have got this, what was the marker pathway that was basically playing a major role? And if there are certain markers that are involved, Are there any specific genes that were involved so that we can tag these markers to those genes? And most importantly, can we use these markers which are tagged to the alleles and develop a marker assisted pyramided line for all the pathotypes? So that's what we wanted to basically look into this. So there were 4 ways of increasing response to selection. The whole idea of a breeder is how he increases his response to selection in the desired direction. You may increase variation by advancing breeding lines of wide crosses through mutation. But more important is how are you going to precisely select? So we moved from a visual selection in breeding to that of a biometric based, what we call as plant to progeny row trials, to pedigree methods, to back cross breedings. We select harder with greater selection differential or selection pressure and grow out of season times. With markers, rapid generation advancement to the speed breeding, all this only increased our precision. So that was I was trying to put. Simple rapid generation advancement, forget about speed breeding. We can take up about 3 generations in field and almost 6 generations in controlled condition. Srinivas Samaneni has done, and Dr. Neeraj Kumar has also done at IRI, so that we can increase the breeding value of a line by having more years per cycle. So I have a greater genetic gain with greater selection intensity. Mind you, I haven't put any marker set here. Having more selection accuracy or genetic variance. So there was already a lot amount of background literature where it was clearly proved that there were specific alleles that were working against each of the races. Race 2 was governed by a single recessive gene. Of course, some earlier studies said 2 to 3 genes. Now we know that further it is a single recessive allele. Similarly with race 3, race 4 and the sort. And to add to our breeding program, over 15 years before I joined in 2007 itself, Professor Shyam Singh Yadav, Professor Behel, Dr. J. Kumar have developed an excellent multi-biotic stress wilt dominated sick plot, and you can see the expression of this wilt in this plot is excellent. Either it is wilt susceptible JG62 or early wilting BGD 1005, they completely vanish, whereas the tolerant lines had. And then we had an excellent screening in this protocol. Till now, I haven't put into any marker-assisted genome-wide scans. A time delay photograph of the same plot of the same location clearly identifies, as I was putting, while we were screening in microplots, while we were screening in hydroponics, I mentioned a few of this line, Latvia ILC0, and you see even in field in a Wilczek plot, it was completely standing resistant. It's not a high yielding line, no doubt, but it was completely standing resistant, where when you look at the background, all those lines that were green have totally vanished. We could develop a large amount of lines. We could work out in the microplot along with that time Dr. Dubey, later Srinivasa, and now Dr. Bag, a large amount of lines which had differentials for various races. But then we never looked at what was the molecular phenomenon, but we were trying to also do some wide crossing using PUSA 372 and Sicer eratinum as well as reticulatum, and this is where we could see that the particular accession ILWC Icarda line wild chickpea 235 was highly tolerant. You can look in field in the Wilczek plot. It was totally tolerant and its derivatives were also tolerant. And because Teresa Milan, during the same time in our Generation Challenge program, along with Professor Rajiv Varshne, who was heading that program, and he was kind enough to grant a good amount at that time when I had no funds, which I am indebted to you, Professor Rajiv, that we could actually identify a set of QTLs which are working. But then we wanted to know what was this molecular, physiological, and biochemical mechanism that provided fusarium wilt resistance in lines like WR315, which was basically a landrace from Kanpur, to that of JG62, which is susceptible to all the races prevalent in India except race 0. And we started looking. So our core research objectives then were, how do you biochemically profile these two genotypes? Priyadarshini has worked day and night both in field as well as in lab, as well as slogging off between plant pathology division, plant breeding fields, and with Professor P.K. Jain from NIPB. We started looking at whether was any phenolics playing a role. Were there any flavonoids playing a role, antioxidant enzymes? Were there any molecular switches that were basically getting on and off when there was an infection? This switch could get on and off. Of course, we found out. We'll be looking into the next few slides. And we zeroed in on to NBS LRR resistant gene and WRKY genes, which were basically the transcription factors. And then we wanted to look where we could establish a correlation between gene induction and downstream metabolic resistance barriers, where they're just housekeeping metabolic barriers or where they're gene induced or transcription induced metabolic barriers. So we had a great experimental setup which worked in sick plot inoculation, which also worked in sick pot inoculation, and later we also did with an hydroponic substantiation. So we had sampling intervals at 0, 6, 12, 24, 48, and 72 hours post inoculation. And then biochemical markers we have checked, as we see of the PAL and POX enzymes. You'll see that the susceptible genotypes in the succeeding thing had shown lower enzymatic activity. But then high induction of the NBS and LRR or WRKY pathways showed delayed or low expression of the wilt symptom even after 72 hours of what you call as pathogen inoculation. When we started looking at phenolics in resistance versus susceptible, there was a good amount of phenolic expressions. If you look at flavonoids, they also have a very huge expression in the resistant line versus that of a susceptible line. And that's what the error bar graphs between JG62 as well as WR315 shows. In fact, it's a surprising thing that WR315 had certain times equal or lower levels of, for example, proline, but then it shot up in its levels post inspection. Immediately, it gave a reaction within 2 days after infection and providing its what we call as anti-scavenging effect. And that's what is clearly seen, where JG62 has a good amount of hydrogen peroxide radicals which could not be scavenged, whereas in the tolerant to resistant genotype, WR315, these were scavenged. Now we started looking at what was PAL and POX reaction. PAL is basically phenylalanine ammonia lyase, which acts as a gateway for polyphenol pathway, the POX pathway. And what you see is within 2 days, 24 hours, there is a peak, and then it gives a rapid activation. Whereas when you look in susceptible, there is some amount of enzymatic trend, but that is not sufficient to counteract the antioxidant, the free radical release, which has occurred in the plant, leading to already damage initiation. So what triggers this? We have done transcriptomic profiling and we have seen that NBS LRR based expression triggers it. The expression is almost 12 times in WR315 when you compare it with JG62. The nucleotide binding, it is not just LLR expression, leucine-rich repeats, but the nucleotide binding site of leucine-rich repeat genes act as a plant surveillance system. So they say, hey, the fusarium is entering, please trigger out and start releasing the scavengers. So in resistant genotypes, these genes are upregulated within 6 to 12 hours, not even one day. Within 6 to 12 hours post-infection, initiating the hypersensitive response from what we call as SARS, system-acquired resistance. Now we have analyzed, of course, DGs at all these stages, differentially expressed genes. So we have seen that among the 59 differentially expressed genes in the resistant genotypes, of course, we have also taken BGD 112, PUSA 256, which was susceptible, and WR 315, and of course, PUSA chickpea Manav. We see that 34 were upregulated, and there were also some 25 downregulated with maximum log fold changes, whereas in some of the susceptible genotypes, only 15 were upregulated. We have identified it to certain master switches, and we had proposed, that has been published in a very good journal, the Physiology PP. The WRKY genes act as master switches, which specifically WRKY16 and WRKY50, which serve as pivotal regulators. You see, what they do in this defense network, they bind with the W box elements in the promoters of defense-related genes, therefore coordinating the synthesis of phytoalexins and lignins. So this is a beautiful classical example. Also provides a future link wherein in the susceptible, but where such genes are there, and due to classical RNA translation machinery or ncRNAs where the function is less, some of the young scientists like you can plan towards even genome editing. But what this study basically highlights is a bi-directional regulations where WRKYs both activate defense and also suppress non-essential metabolic pathways during infection. So defense cascade is integrated by this master switches. What they do? They first sense to the plant that pathogen-associated molecular patterns, PAMPs, are recognized by the NBS, nucleus binding site leucine-rich, this thing. They signal WRKY transcription factors, translate recognition into a transcriptional response. They immediately say, oh, you start transcripting proline. Further, they form a barrier because they drive phenolic and lignin deposition. Therefore, they try to form a barrier in case before the wilting starts. And at the same time, what they do? They actually activate scavengers, superoxide dismutases, which actually manage the hydrogen peroxide and other reactive oxygen species. You see how beautiful the mechanism is, right? That they sense, they signal, they form a barrier, they detoxify. Perhaps one of the first studies of how genes integrate into signaling pathways in case of wilt. We are still now working on it. So what are the key takeaways from Pridarshini's research? That there is a dual layer of resistance that is basically working in the resistant genotypes. The resistance is achieved through both strong physical barriers and rapid molecular signaling. And that's where Raupreet is now working on, one of my PhD students as well as a research fellow with me, where she is trying to understand whether these are the genes or whether they're influenced by the mate family genes. How are mate family influencing these master switches? She is looking into that. The genetic selection of NBS, LLR, and WRKY genes are confirmed as high potential markers for marker-assisted breeding. The earlier markers that we could identify. There is a sustainable management that because if you develop cultivars with these traits, We can reduce chemical fungicides. We provide a boost to organic cultivation. And most importantly, the pulse farmers are marginal farmers who grow them on marginal lands without fertilizers, without basic inputs of pesticides, and this will become a what we call as renaissance for them. Can we predict activity? Because their POX activity is within 6 to 24 hours. Maybe some of my physiological friends, physiology friends and the pathology can also develop some screening tools or ready-made kits for the breeders. Now we had very good information on the phenotyping. I'm sorry, gulping a little bit of water. We had a very good information on genotyping, the markers assisted, and this was the time when the Generation Challenge program attached us with Professor Varshney's lab, Manish Rurkiwal at TICRISAT. They had a DBT program, and I was made as an ex officio partner with it. And then we screened a large amount of lines, a large amount of markers, developed very good MTAs, identified the narrowed down on that particular loci, and also could actually identify the genes responsible. We thought that now, since there are fork alleles, can we start looking at pyramiding? So what was the breeding bottleneck? For example, we had an agroeconomic core in PUSA 391, PUSA 372. For example, even in case of Dr. Aditya, who was working at IIPR Kanpur with PUSA 256. Now, the lines that we were working on at IRI, where Dr. Jorben actually initiated all this program, of course, crossing was done by the genetics chickpea unit, that we started looking at PUSA 391, which was a mega desi chickpea variety when it was released. It was having very stable yield across zones and locations. But then slowly it dropped down because it became susceptible to wilt. Why? Because the pathotype concept has developed. There was a pathological shift. Increasingly dynamic strains of Fusarium oxysporum forma species Siserae have recently overcome its defenses, inducing massive wilt outbreaks. Therefore, our aim was, can we use the marker-assisted backcrossing to stack resistant genes without losing the PUSA 391's core elite agronomic profile? So here we had a desi variety, but we wanted precise backcrossings than just generating backcrossing, then generating huge amount of lines, wait till we could screen them when the family character at F5 or F6 started, and I can take them to my Wilczek plot. And we had a donor parent, well characterized for the fork races, well identified against 1, 2, 3, 4, 5 fork except for 0. It had a target resistant haplotype in the genes identified, and therefore we had before us a spectacular molecular breeding scheme. But most important of the scheme was we had excellent collaborative team of the CEG lab at ICRISAT, breeding and phenotypic partners at IRI, Professor S.C. Dubey, later Srinivasa, now of course Bagh, similarly at IIPR Kanpur, good collaborations and trustful collaborations between us partners, and we started to have this base by my friend from Colombo University, Professor Madam Teresa Milan, who way back published, and I joined in 2007 into the chickpea program, and I had this gap analysis where a detailed map with this target genes was there. So when we screened a large amount, though TA 110 was there, it was not showing a lot of variation. So we could zero in into GA16, TA21 and TA96, which were selected for foreground selections to ensure successful target gene introgression. And across we had 8. So we thought that at least we have 6 distributed evenly chromosome. Now we have moved to 10 for the recovery of genotype. And most important, along with the CIMET, Abhishek Rathore, thanks that we developed a breeding pipeline where we had a BC1F1 selection, BC2F1 target, BC3F1 focus, and BC3F3 trials. So we had a cross where we evaluated selected plant showing 75 to 80% parent recovery, but at the same time for the foreground selection, of course, since it's a BC1F1, it will show not homeotic but heterotic combinations. So such heterotic combinations were used as male because we want to recover more of the female parent. So we use the female parent as the donor recipient line and the F1 as the male. We started crossing to have a 90% recovery in BC2F1. Certain times it was sufficient, but then we wanted to be doubly sure and got through BC3F1. So one season at IRA main station, second, thanks to my close friend and colleague, Dr. B.S. Patel, where we had done at Dharwad, then again at IRI. So we completed in 1.5 years, and then we started from BC3F3, BC3F2, BC3F3, BC3F4. So we started using ICRISAT off-season trial to that of Dharwad, so that in the main season we came back. So this has completed 1 whole year and 1 more year at Wilczek plot 2 years, and 3rd year our lines were ready to go down into multi-location trials. So 14, 15, 16, 2 years of developing, 2 years of testing, 4 years, and immediately pumping it into ACRIP trials. But before that, because this was our first strong, earlier there was one variety that was released along with Pusa chickpea 10216, which I have molecularly bred for drought and Annigiri, but that was only for FOC2 for the southern state. And this was for multi-races, and excellent work here with Jorben and all our friends across the locations, including Siddharna Gowda Patel, Madam Jayalakshmi, and all, where they have screened across the locations using the NDPI, found out the most stable introgression line IL1 and IL4 in this quadrant, which we could clearly identify. We are now sitting on a gold mine of lines, almost with 5 markers, polymorphic background 98. As I said, I moved to 10 to 20. We have used for background selection, MABB lines, we have 61. Derivated common trial 6, but we have the limitation of their ability to enter, and now it has become almost an academic exercise because the pathogen load in the Wilczek plots need to be characterized. But already what we have done, that we could develop huge amount of lines which are having very high yield, very high resistance with good amount of seed weight, and both of them doing excellently well. So now we have evaluated this across locations, and as I said, IL1 and IL4 in Jorben study, which is BGM 20211 and BGM 20212, were identified the IL1 and the IL4 as the most stable lines in the lower left quadrant, and we have pumped them into ACRIP trials. And once we have pumped them into, you could look either it was PUSA 372 or the other lines. When they were pumped into the ACRIP trials across the wild specific locations, even against JG315 or even against Annigiri or our lines, both 20211 and 20212 have out-yielded all the best checks as well as leading lines. So it was Pusa chickpea Manav, BGM 20211, was the first fork allele pyramided QTL introgression line ever released in the world, which has a whooping 28% yield advantage. You can imagine it has averaged 3 tons per hectare, has a potential of 4 tons. Farmers are taking even 6 to 7 tons. Grain protein content 18.92, Released in 2020, today it has 470 quintals of breeder seed in it. A mega variety, which not me, but agricultural minister has said, show me your super chickpea variety. Where do you have it? And this was only possible because of partnerships, because of collaborations, because of trustworthy collaborations, whether it was ICRISAT, whether it was IRI, whether it was IIPR Kanpur. We worked in tandem. Excellent lead by Professor Rajiv, Manish Rurkiwal from IIPR Kanpur, ICRISAT, with Anu Chitkineni giving all the HTPG support to us, and excellent farmers, whether it was Lodi, whether it was Shinde, whether it was Anil Kate, where you can see the fields with high number of this thing. As I said, we had before one more variety, Super Anigiri, but it was only for FOC 2. Now you can look, these are not single plots in my Wilczek plot. These are large lines of 200 meters length, showing absolute resistance compared to that of the susceptible check, to compare that of the breeding lines. So now we are moving, can we use genomic selection approaches to develop super chickpea varieties? That means those which have an average yield of 3 tons. You see chickpea wherever ACRIP has 2.4 to 2.7 tons with a potential of 2.8. We are looking at averages of 2.9 to 3 tons with potentials above 4 tons. We have released, thanks to Dr. Sailesh when he was here, that we identified Pusa chickpea Ashwini, which has a 3.8 tons production potential with average again of 2.9. So the genomic selection strategies, maybe some other day I should be looking on, but with the decreasing genotype costs, to that of making selections of target years. Now with rapid generation advancement or speed breeding and using marker trait associations, having greater favorable alleles due to the genomic selection, reduced years of cycle, we can always have greater genetic gains. Now, if you look at this traditional breeding, this is my last slide. The traditional breeding of a release of varieties where we take 10 to 11 years, the marker assisted breeding and the genomic selection have reduced this to 6. This is without speed breeding. This is with only rapid generation advancement of off-season and using a third crop at ICRISAT. Whereas the marker-assisted breeding, the number of crosses that can handle is very less. But with that of genomic selection, the funnel mouth is very wide, but it rapidly narrows down to those genotypes or the haplotypes that are carrying the favorable alleles, and then can initiate a second cycle of selection, further narrowing down and then selecting the best. That's what, friends, is the power. That's what I was saying here. I'm very much thankful. Again, I always put the slide and this young short Sunil Gavaskar of pulse breeding, Professor Rajiv Varshne. You can look 2.12 from Pusa chickpea to 10216, 4005 to 3.195 tons per hectare in Manav. An excellent genetic gain. It was such a valuable thing that when Gates visited Latur, of course, this was with the drought-tolerant varieties, he was wonderstruck that all chickpea failed except for this cultivar, which he inquired and said to the PMO, I would like to visit these fields in IRI. And clearly he has mentioned in his paper thing that of the 3 technologies that I believe that can be done is number 1, medical, another is fuel, and third is the next generation chickpea plants currently growing at the Indian Agricultural Research Institute, which are a staple crop of India. And he says that I'm stubborn in my belief that with the right innovations and delivery channels to get them to the vulnerable, we are capable of making progress across not just India, but across the world. Basically, friends, this is not my work. It's a synthesis of work of my team, which includes my staff, Dr. Neeraj, Dr. Rajesh, and of course, Patil is missing in this scenario. All my previous and current researchers, Dr. Jorben, Dr. Apurva, Dr. Supriya, Sneha, now of my postdocs, Dr. Gayatri, and we have Dr. Kapil Gautam, and my research students, Jyoti, Ken from Myanmar, Rupa, Raupreet, who is working to look at the mate analysis, and Uma Shankar, who is working on aluminum toxicity. And of course, we are missing some of the students are here. Mr. Bharat, who is working on phosphorus. I think we have Nilesh, we have Sumit, we have groups, and most important, it is collaborations. Whether it was IIPR, NIPB, the national partners like NIPGR, Kota Agricultural University, the Punjab Agricultural University, Benares Hindu University, Andhra Angrao, or my international partners, the Excellence in Breeding platform from CIMET, my partners Hamavi Tapan, Professor Shiv Kumar from Mikarda, my excellent partners Rajiv, who has moved to Murdoch University, Professor Kadambot Siddiq at the University of Western Australia, the Institute of Agriculture, Himabindu, Rakesh Srivastava at ICRISAT. Most importantly, my funders, my parent organization, IRI, under the umbrella of ICAR, Bill and Melinda Gates Foundation, or the DBT program. I very much thank you, Ankur, Madam Pragati, and whole of the team at Ankuran 2026, who have conceptualized this 30-day summer school, gave me an opportunity. I'm open for discussion. Thank you. I'm stopping the share, and please do ask.
Thank you so much, sir, for sharing such a valuable knowledge and insightful presentation with all of us. And now we'll have doubt clearing session. So dear participants, if anyone have any query or question, kindly feel free to ask them directly to Dr. Bhardwaj sir by unmuting yourself, or you can drop your doubt in a chat box to read. The chat box is open to all.
I have a problem. You need to read Revangani chats. Somehow today, first time if there are any in chats.
डॉक्टर साहब ये देखिए आप कि 250 पार्टिसिपेंट ने यहाँ जूम पे ज्वाइन किया था, एक सिंगल कैंडिडेट ने अभी तक लेफ्ट नहीं किया. इतना इंटरेस्टिंग टॉपिक.
थैंक यू.
और यूट्यूब पे आप देखिए 200 प्लस पार्टिसिपेंट, इवन 500 प्लस लोग इस लेक्चर को सुन रहे हैं.
Thank you, sir. Thank you. Now, that is because I have seen not only in this, in the earlier program also, that you have a very good reach, maybe because the content of these trainings is useful. I have a question from Ajay. How can breeders ensure resistance remains effective against emerging pathogen races? This is an excellent. That is why you require, an effective screening tool. If you don't have an effective screening tool, you will not be able to work and identify what are the emerging pathogen races. Second, that is why I said team and collaboration. Unless a pathologist identifies a differential which is resistant or tolerant against a race, a breeder is useless, because then he'll be hitting in what you call dark. So a pathologist provides a target. This is the race that is becoming dominant. A pathologist provides the bullet. This is the differential that is resistant to the race. A germplasm mine actually gives him the large number of germplasm lines that a pathologist identifies. A biochemist and a physiologist identifies the resistant mechanisms like it was done here. PK Jain, Vinuta, then we had Ruchi now with us, and our own phenomics man, and a biotechnologist identifies the MTAs or the genes that are playing. These are the ammunitions that you have to use. These are not the things that will replace a breeder, but they will help the breeder in better targeting and precision breeding. Unless a pathologist identifies what are the emerging races, breeder will be going down with the old ones, right? So it's a teamwork. Hello, sir. Myself, Nikhil. Want to natural farming? No, Nikhil, that's a big question. We have Pusa Green 112, Pusa Chickpea Manav, Pusa Ashwini, which go excellently under natural farming. We have Professor Dinesh Kumar from the Division of Agronomy. Most importantly, our Assistant Director General, Professor Shive, who has become Assistant General Director General HRD, is an authority, and Professor Dr. Kapila Shekhawat. Pusa Green 112 does excellent. Hypocholesteremic, sir, that is reducing diabetes. क्या चने में बीटा साइकोस्टेरॉल है? I think this is a different part of a lecture. There are many provitamins, vitamins, beta carotene, and large amount of nutrients Dr. Neeraj is working on to identify marker trait associations. Along with Professor Rurkiwal, we have identified through Jivas. Not only that, we have recently released lines. For example, Pusa Chickpea Ashwini has a protein percentage of 24.8. Our Pusa 4035, the world's first double dollar Kabuli variety, has 24.5% protein, whereas you have others which are at 22 to 23. And now through genomic selection, Neeraj is working on to increase this protein percent to 26. We have already submitted in the protein nursery trials lines having 25.5 coupled with higher yield. You see, earlier everyone thought there was a negative correlation. Now we are trying to break it, bring in the haplotype breeding to increase it to something unimaginable. Just our next 5 years target is to bring it somewhere around 25 to 26. So that will be our target. Sir, this is one of the way Samsung SM, it doesn't have any impact. It's not a transgenic variety. A normal आनुवांशिकरण यानी normal क्रॉसिंग से बनी हुई वैरायटी है, ये कोई ट्रांसजेनिक वैरायटी नहीं है. तो इसमें कोई सॉइल में इंपैक्ट ऐसे नहीं है. रादर इसमें गांठ बहुत अच्छे बनते हैं. राइजोबियम फॉर्म्स वेरी गुड. वन ऑफ माय स्टूडेंट इज नाउ लुकिंग कि व्हाई द विल्ट resistant variety is able to allow only rhizobium but prevent fusarium. Now, of course, Parichita's research has given a beautiful direction that within 6 hours, that it is first of all doing two things. It is preventing the fusarium to enter into it. You see, now it doesn't prevent that way. So there is some amount of microbiome activity that is going on. With Professor Varshne and group, we are coming out with an excellent paper with Central University Hyderabad. But at the same time, Professor Neeraj, Dr. Neeraj is working on to look at this dynamics with Swarna Lakshmi. Beautiful. Wild chickpeas, whichever are crossable, can be used. I have given a lecture at PGTSAU. for base broadening as well as genetic widening. Now you need to understand that pre-breeding itself is a very complex activity. They certainly base broaden as well as widen the genetic base. But then so much of variation, so much of lethals is thrown out that Professor Sandhu Saab, who rise to the height of DDG in ICR, later as Vice Chancellor at 2 universities, has spent his whole career, I call him the chickpea wild species breeder, whole career on this, and he has provided with PAU and us an excellent array of lines. But then it takes a lot amount of actually bringing them into cultivated lines. It's a huge process. Even after 20 years, these wild species throw out good characters for their survivability, but not characters suitable for farmers incentivizing. So you need to incentivize pre-breeding. Gayacharan is working on it. There are many other compounds that work by. Has any formal hormonal profiling on the wilt affected? Yes, now Dr. Vinita is working on it and it's in the preparatory stage of the paper. Yeah, I think I have completed all the questions on chat. Anyone want to raise your hand and then ask? I'm free for it.
मुझे नहीं लगता कि सर अब चैट बॉक्स में कुछ बचा है।
नहीं, चैट में नहीं है, so I'm just
हाँ, लेक्चर ही अपने आप में इतना सेल्फ एक्सप्लेनेटरी रहा और जो डाउट्स थे पार्टिसिपेंट्स के उन्होंने पूछ लिए। सब लोग आपका धन्यवाद ज्ञापन कर रहे हैं।
बढ़िया सर, आई थिंक वी कैन क्लोज।
जी जी सर, ये हमारा पहला नहीं, भगवान के पास ये शायद छठवां सातवां अनुभव है। ये हमेशा सबसे बेहतरीन लेक्चर रहा।
नहीं, बढ़िया सर, एंड ऑल द
और
पार्टिसिपेंट्स।
मेरी इच्छा थी सर कि पहला लेक्चर आपका ही हो, आपके लेक्चर से ही मतलब सीरीज स्टार्ट हो, है ना?
थैंक यू सर।
डॉक्टर रेवांगिनी।
Yes, sir. So, as we come to the end of today's session, I, on behalf of entire organizing team and all the participants, express my heartfelt gratitude to our esteemed speaker, Dr. Bhardwaj sir, for such an insightful, inspiring, and knowledge-enriching session. Sir, I personally believe that all the participants greatly benefited from your talk today and gained a deeper understanding of this subject. Your scientific vision, motivational words, and remarkable contributions have inspired every participant to think beyond boundaries in this field. I would also like to sincerely thank all the participants for their active presence and enthusiastic participation throughout the session. We hope today's interaction become a source of inspiration, learning, and motivation for your future journey. With this, we come to the end of today's session. We hope to see you again in the future sessions. Thank you to all once again. Thank you so much, sir.
Thank you. Someone was asking, we get short notes. Ankur has clearly indicated it's there in YouTube. You can always access it. Thank you for the beautiful words, Revangini. I think I pronounced
बहुत
it
बहुत
right.
धन्यवाद सर, बहुत बहुत धन्यवाद। जय श्री राम।
Jai Shri
आपको
Ram,
बहुत
Jai
बहुत
Shri
धन्यवाद।
Ram. I'm logging out. Thank
जी
you.
सभी, जी सभी पार्टिसिपेंट्स लेफ्ट कर सकते हैं। थैंक यू।
Thank you, sir.